Good afternoon, CGIL. This week, we're very happy to have Dr. Gonzalez-Recio presenting for us from the Rosalind Institute. Dr. Gonzalez-Recio completed his PhD in animal breeding in 2006 from the Polytechnic University of Madrid. Then he spent his postdoctoral period in the University of Wisconsin in Madison, working on statistical modeling of genomic information on complex phenotypes. Since then, Gonzalez-Recio has obtained international experience as a senior research scientist in the Department of Environment and Primary Industries in Australia and the Department of Animal Breeding at INEA. He is now a professor at the Rosalind Institute with a personal chair on livestock hologenomics. His most recent research projects include the selection of feed efficiency in lower methane emissions, including the role of microbiome to breed more productive livestock that provide more food for human consumption with reduced use of raw material and low environmental and land footprints. Dr. Gonzalez-Recio emphasized the importance of phenotypes and recording schemes to maximize the success of breeding programs, as well as to improve the inference of prediction to be made for a better understanding of genetics and biology. So I'll turn it over to now Dr. Gonzalez-Recio, and we can get started.
Thank you, Julia. Thank you for the introduction. I'm very glad to be here with you in this seminar, webinar for me. The title of my talk is Breeding for Lower Methane Emissions in Cows, Current State and Future Plans. I was talking like, I think that 2 or 3 years ago there in Guelph about our plans and our current research. So I think it will be a good idea to update what we have been doing during this time and what are our future plans. So this is what I'm going to talk about today. Why are we interested in methane? What is our current projects? The new trait that we are defining and how we include methane emissions and feed efficiency in the economic profit functions that we are developing and we have already published in Spanish dairy cattle, and also how to include the microbiome in microbiome-driven breeding. Let's start with methane, and I will start with the limitations of methane emissions in dairy cattle. Selecting or breeding for lower methane emissions puts a lot of pressure on farmers without benefits for them, because so far they are not getting incentives or penalizing for emitting methane. And it is actually true that it accounts for a large share of greenhouse gases emission in agriculture, about 40 to 50%. But it's only a small proportion of total greenhouse gas emissions. Worldwide is between 5-6%. And mitigation potential is limited for biological reasons, because this methane comes from the enteric fermentation of feed, and that's something that is a biological thing of the rumen. So there is a limited mitigation potential of reducing methane emissions because ruminants are going to emit methane. However, short-term mitigation for a rapid decrease is beneficial for global warming, because methane is a short-life carbon that lasts only between 12 and 20 years in the atmosphere, and it has a warming potential much larger than the CO2. It is also a loss of energy from feed, so it's an economic burden for farmers, and also it's an opportunity for the livestock sector, for farmers to say that they engage with the society and they are committed for sustainability and mitigate global warming. So if you have listened to me before, you know that we are working in measuring methane since 2017. We're using sniffers in commercial farms. By now we have measured actually almost 5,000 cows, and we have also MIR records for some of these cows. Since 2024, we were involved in another project that is called Dairy One Holistic. Before I moved to Scotland, but I still collaborate in this project. We are measuring intensive and organic farms using green feeds. And in December last year, in December 2025, we already had 200 cows measured with these devices. We're also registering MIR for all these cows. And you can see some of the pictures here, how we set the green feed in the farms, and you can also see how we set it in the organic farms. Spain is not always sunny and dry. We have very humid and cold regions, mainly in the north of Spain, as you can see in this picture. We have a genetic evaluation, official genetic evaluation since 2023. And December last year, we got a new definition of the trait that I'm going to explain today. And this is the one that we're publishing right now. And in Scotland, we are working on measuring methane with the laser methane device. This is a handheld device that you probably know already. Here's Luna measuring methane in our cows. And we're also installing sniffers, in this case the mulogger in the farm. And we're also going to sample the microbiome, which is an ongoing project within the Global Methane Genetics Initiative, and in particular, this microhub project within this initiative. I was going to talk about how we process the data once they come from the sniffer and the automatic milking system with the robot, but there's going to be a specific session very soon. I cannot remember the day, but it's going to be a specific session within the Global Methane Genetics Initiative on this topic. If you're interested on this, and I think that somebody from Guelph is also participating on how we process the data files from the from the sniffers and from the milking robots. So I'm going to skip that part, and I will go to the new phenotype definition. We're interested on converting ppm to grams per day, so concentration to production. You know that the sniffer only measure ppm, so this is concentration in the air. And most of the equation right now that try to convert concentration to production uses this ratio of methane to carbon dioxide. We can obtain this ratio in the sniffers using two smart cards, that one measure methane concentration and the other one measures carbon dioxide. So on this new phenotype definition, what we do is to calculate the expected carbon dioxide emission per cow per each comparison group. And we use this equation from Pedersen et al. In 2008. Is the same equation that Madsen used, and it basically uses the heat production units, which are dependent on body weight of the cow, energy corrected milk, and days in pregnancy. So this is an example, for instance, of a comparison group, and this is the expected distribution within a farm and a period that we are measuring. Then what we do is to estimate carbon dioxide from the sniffer samples, and this actually depends on the sniffer flow and the cow breath and the concentration of CO2 that we are able to collect. So we calculate this. We do an estimate, a daily estimate of the CO2 emissions from the sniffers, and actually you can see that these two distributions they overlap, but they're not the same. So the CO2 estimated or the expected one, which is the light brown, is different from the one that we observed in the sniffer. So what we do here is to transform the distribution from the observer samples to the expected one. So this is basically a transformation of the distribution, just to put the observed one, the observed CO2 emissions or concentration in the expected distribution of CO2 in that comparison group. Once we have this will be the observed carbon dioxide production for each of the cows in the expected distribution, and we use the ratio between the carbon dioxide and methane to estimate methane production in liters per day or grams per day, because then we can use a density of methane to convert from liters per day to grams per day. And we have compared this new trait with all the other traits that we were measuring before. And here you can see an example of a visit to a cow in a robot. It lasts 5 minutes, so we take into account only the first 5 minutes of the visit. There are 3 peaks here, 3 eructation peaks. And these are all the traits that we have measured, or we have defined. We calculate the average methane in ppms, which will be like this value here, this horizontal line. We can also calculate the average value of methane emissions, but taking into account only one measure every 5 seconds. We know that this is the case for many of the sniffers, or many groups are using only one measurement per 5 seconds. The area under the curve is just the sum of all the values, so it will be this trait here, the light blue. And we can calculate also every second or every 5 seconds. We can calculate also the maximum peaks, which is, these are the peaks. You can see that we have 3 peaks, and we have the value, the maximum value of each of these peaks. Actually, what we do is to average the 2 largest value for each peak here. So this will be the value of each peak, and then we average them and calculate the value per minute. Number of peaks, again, we have 3 peaks, and in 5 minutes, so we calculate the number of peaks per minute. In this case, it will be 0.6. And we also calculate the average value for CO2. This is not in the picture here. We calculate the ratio between the average methane value and the average CO2 value. We calculated methane production in grams per day using Madsen equation, and also this is our new trait, which also calculates the expected methane production in grams per day using the equations or the procedure that I just explained. And here you can see the heritability estimates and genetic correlation estimates about the diagonal for each of the traits. You can see that most of the traits have had a heritability close to 0.10, except number of peaks per minute, which had a larger heritability. You can also see the genetic correlations about the diagonal, and in general, we can see large and positive genetic correlation, except for the number of peaks per minute, which was depending on the trait. It could be moderate positive or moderate negative. In the reliability, the repeatabilities for the traits were high in general, between 0.6 and 0.75. And I would like to highlight here 3 genetic correlations for the new trait that we are using. It had a large genetic correlation with the average of methane, also with the ratio, and also with the methane production calculated from a Madsen method. In the genomic evaluation system, this is what we use. Our trait is actually we implemented a bivariate single-step GBLAB. The two traits that we analyze is methane concentration from sniffers. Many of the cows recorded with version 1 of sniffers when we started to measure methane, they do not have a CO2 concentration, so we use this trait for those animals. For all the others, we add this other trait, the pretty methane production. This is the trait that Spain is officially publishing right now. Okay, so but the model that we're using is a bivariate single-step GBLAB. As systematic effects, we include lactation number, days in milk, and month of calving. And for random effects, we include hair robot week as a comparison group, the permanent effect of the animal, and the additive genetic effect of the animal, plus, of course, a residual effect. This is the number of records that we had, almost 20,000 records for methane concentration, and more than 25,000 records for this new defined methane production trait. The heritability of the trait is 0.12 and the repeatability is 0.55. Again, for the trait that we are publishing right now in Spain. The genomic estimated breeding values were published before in a scale that was normal with an average of 100 and a standard deviation of 10. Since June 2026, CONAFE is going to change the definition of the trait, and it's going to either express the trait in units of emissions in grams per day or in potential reduction of emissions with a base of 1990. This is because most of the inventories, greenhouse gas inventories, set this base in 1990, and they are interested on calculating what is the potential or what are the emissions in grams per day. So we think that this is going to help to the inventories, and this is going to also help policymakers to pay more attention to breeding to breeding for methane emissions and understand better what is a genetic value and how they can decrease or mitigate methane emissions. This is the genetic trend for the methane trait, and also for comparison purposes, I'm going to include feed intake. You can see, and this is something that we have seen and I've told before, we cannot observe like a clear genetic trend for methane. You can see that is basically quite flat. And this is as I said, this is a new way to express the trait in terms of potential of reduction of methane emissions compared to the population of 1990. Some sites have a genetic potential of reducing methane emissions 10%. Some others will increase methane emissions by 10%. In PTA, this will be half. Okay, but in general, most of them will be between -5 and +5%. This is different from dry matter intake, where you can see that the genetic value or genomic values has decreased. So this means that the current population eats more genetically, are more prone to eat more feed than the generations before. So Everybody's working on large-scale methane phenotyping, but this is still a challenge for many countries and for many production systems. Sniffers are mainly installed in farms with milking robots, or otherwise they have a low throughput. On the other side, green feeds are expensive for large-scale phenotyping, and other devices like SF6 method or the respiration chambers, they're not feasible to implement at the commercial level. So we're trying to find proxies or other traits that can be included in the breeding programs. And I know that many other proxies are proposed. We're working with the microbiome. Why microbiome? Because we have seen in several studies that it is genetically correlated with methane. It has a genetic correlation larger than 0.70. It shows heritabilities moderate to quite high for what we are used to get, between 50 and 40%. And in this study by Sepúlveda et al. In a collaboration between Spain and Australia, we saw that we could increase the response to selection for methane up to 50% when we include both sources of information, methane emissions and microbes. It is also correlated with digestion efficiency, so having microbiome could also be a good proxy for feed efficiency. And we can think that selection, if there is a genetic correlation between methane and the microbiome composition, and also between the feed efficiency and microbiome composition, it's logic to think that selection against methane and also in favor of feed efficiency may change rumen microbial composition. And this may impair feed efficiency, or it may impair rumen health. So if we're going to select for a trait that is so highly correlated to rumen microbial composition, and we know that the rumen is a very important organ for ruminants, we need to know where are we driving the rumen microbial compositions to using selection. But we also agree that large-scale phenotyping for the microbiome is still a challenge. So there are some research questions to solve this that are being currently undergoing, like, okay, what if we could use other microbiome communities like the buccal swabs or use genomic selection using a reference population together with methane and feed efficiency, if this could be enough for obtaining a good genetic progress for these traits. So in our microbiome-driven proposal, we are using a population of 450 hosting cows in first and second lactation that were in 14 farms. And we use whole metagenome sequencing using long-read Oxford nanopore technology sequencing. We use aggregated microbial variables that are defined as in Saborío Montero et al. 2021. Basically, what we did is to, after the CLR transformation, to account for the compositionality nature of the metagenomics data, we use principal components, and we obtain two aggregated variables, which are the first principal components and second principal components, but we can, of course, use more principal components. But this too has quite clear biological meaning. And for instance, principal component, the first principal component is associated with the percentage of eukaryotes in the rumen. We know that the eukaryotes live in symbiosis with archaea and methanogenic microorganisms, and they are involved in a different hydrogen utilization pathway. So a larger proportion of eukaryotes in the rumen is also associated with larger methane emissions. Whereas the second principal component is associated to this ratio here, proteobacteria divided by firmicutes plus bacteroidetes abundance. And this is also related with acidosis and microorganisms that have a quicker degradation of fiber, and they may produce acidosis in the rumen. I didn't put the heritabilities for these principal components, but they are close to 0.20. And you can see that they are correlated, both taxonomy. It doesn't matter if we use taxonomic annotation or functional annotation, the genetic correlation between them is quite high. We estimated the genetic correlation with other traits, but I will highlight correlation with fat yield and actually also with milk yield and protein yield. They have low but quite important genetic correlations, mainly with fat yield. You can also see that the genetic correlation of some of the aggregated variables with fat yield is high. You can also see that the genetic correlation with methane production and with dry matter intake is quite high with these aggregated variables. And also some of these principal components are also genetically correlated to the composition. This is the capacity index. So this is the body composition, body weight, chest width, size, etc. So it's a compound index of capacity and size. So you can see here that larger cows have a different rumen microbiome composition than smaller cows. As summary, ruminotypes with a larger proportion of eukaryotes in general. Sorry, this is the other with lower ruminotypes with lower amount of eukaryotes produce less methane, have a lower dry matter intake, and they have larger production traits. Smaller cows, so low cows with lower capacity values for the capacity index, apparently also host less amount of eukaryotes in the rumen. This is probably because the rumen is also smaller, and the feed transit is quicker, and eukaryotes do not have so much time to grow and degrade fiber, the less degradable part of the fiber. So with all this information, we have developed new economic selection indices for CONAFE in Spain. These are purely economic selection indices using profit equations to calculate the economic value of the traits. You can see here what economic values we have calculated for each of the scenarios. So depending if we are working in a traditional intensive, or we're working on farms that are more focused on cheese production, farms with more grazing systems, or farms that are organic farms. You can see here what are the economic values for these traits. I don't think that we need to take much in detail on this. I will just mention the huge difference in the economic value of methane production. We use the value or economic value for methane that was based on voluntary market carbon markets. So since they are voluntary market, the prices are much smaller than when we use organic systems. Here, what the value for methane that we considered was the expected value of carbon projected to 2050 in the regular market. So this is much a largest price for carbon. Just to highlight the difference between the economic value for methane in these systems versus the organic one. Why we use the voluntary market for these farms? As I said before, because so far there is no incentives or penalizations for selecting against methane. So this is just a voluntary breeding goal that the dairy industry is willing to do. These are the weights of the different traits in the indices, depending on the benchmark. And in the benchmark is what we use is, okay, we're not selecting for methane production or dry matter intake. This is the current scenario for these economic weights. And now in the new economic selection indices, in this EMET, we are including methane production and dry matter intake. So you can see that now these two traits appear with some weight in the indices. Again, a lot of numbers. I will just focus on production traits and the most two different systems, which is the intensive, traditional intensive, and the organic system. Grazing and cheese focus are something intermediate. So you can see here that the EMET, the new EMET in intensive systems, with the inclusion of methane emissions and dry matter intake, the weight of milk, fat, and protein gets reduced in the index, and the same for the organic system. And the reduction is much larger for the organic system than for the traditional intensive. This is the weight in which methane production and dry matter intake are incorporated in the index, 1.6 or 2% more or less for methane production, and almost 14% of dry matter intake with negative values, so selecting against methane production and against dry matter intake. In the organic system, the weight for dry matter intake is similar, around 14% against dry matter intake, and of course much larger for methane production because as I said, this the economic value for methane in this system is much larger. So it achieved we calculated an expected weight in the index of almost 12%. This is how the traits changed with the new selection indices. You can see that the traits that had a larger variation was longevity, when we include dry matter intake and methane production. Of course, methane, because this is a new trait, and also dry matter intake, because these are the new traits that we are included in the selection index. But you can also see reduction on the weight that we put on production trait, milk, protein, and fat. When we compare the traditional intensive versus the cheese focus, the grazing and the organic, you can see also what are the traits that are that change more between these selection indices or these scenarios. In the cheese focus index, you can see that protein and fat are the ones that have a larger values in comparison to the intensive system, traditional intensive. Grazing and traditional intensive, they look alike quite much, very much, except for protein, in which grazing has a lower relative weight. When we compare organic to intensive system, you can also see that methane is the one that have a larger weight in organic, also protein and fat for these traits. Everything else doesn't change that much. And we have also calculated what will be the expected genetic response if we include these aggregated microbiome variables that I mentioned before. So in these scenarios, we are going to assume that methane and dry marine intake cannot be measured, as is the case of for many farms or for many systems. And instead, we assume that we can get a microbiome sample from each cow at least once during their lifetime. So we evaluate microbiome instead of dry marine intake and methane, because as we saw in the genetic correlation table, they are very strongly correlated to each other. So this is what happened when this is the results, the expected genetic response when we use dry marine intake and methane emissions, like in the indices that I mentioned before and that I shared with you. And these are the options when we replaced methane and dry marine intake for the microbiome. And two scenarios depending if we use taxonomical annotation or functional annotation. Basically, what you can see here is that the expected genetic response in milk yield, protein, and protein yield decreases when we include the microbiome aggregated variables, but we increase the expected genetic gain for fat yield. And this is because, as I showed before, there is a larger genetic correlation with microbiome composition and fat yield than with the aggregated the microbiome with milk yield and protein yield. And fat yield is actually a trait that is antagonistic with methane emissions. So apparently, the microbiome is doing like the microbiome variable is acting as a buffer variable so that we don't need to decrease fat yield as much as when we just use methane emissions in the trait in the index. Everything else is quite similar. Again, excepting for methane production, where we can achieve a much larger reduction using the microbiome information. And this is in agreement with the paper that our Australian colleagues published last year, also that we can do a much better job using microbiome than using methane. In terms of profit, you can also see that using the microbial variables, the profit was actually increased in comparison when we used methane production and dry marine intake. So this is something that is quite positive, it's quite encouraging, and we could use microbiome variables for selecting against methane emissions and dry marine intake. This is the scenario again for the other most different production system, which is the organic ones. Here you can see that the genetic gain for milk yield is more or less the same, but again with a little bit of reduction when we use microbiome information instead of methane and dry marine intake. Also for protein yield, and actually in this case we are decreasing protein yield. But on the other side, we are increasing the genetic gain for fat yield, where in the scenario or when we select or when we used methane and dry marine intake, we were actually decreasing fat yield. Again, other traits remain more or less similar. And if we go for to see what happens with methane production, again, we do a much larger reduction of methane production using microbiome information in comparison when we use actually direct methane measurements and dry marine intake measurements. Again, the profit expected genetic gain was larger when we use the microbial information in comparison when using methane and dry matter intake. So this is a very preliminary results. It is under review in Journal of Dairy Science, but it's quite promising, and it is in agreement with previous results from other colleagues. So as a take-home messages, methane phenotype, we are proposing a new phenotype from sniffers that is expressed in grams per day, because there is quite a lot of interest on expressing this trait in grams per day. It does not depend on individual production traits, and it is highly genetically correlated with other traditional phenotypes. So this is the one, this new methane phenotype is the one that is going to be published in official genetic evaluations in Spain after this summer. New economic indices that include dry matter intake and methane as breeding objectives are being developed, purely economical indices, and they achieve better genetic response for both feed efficiency and methane emissions. And again, this is well, actually, these images are currently published from CONAFE, and they are available already for the farmers. In terms of microbiome-driven breeding programs, what we see is that microbiome is a good alternative when dry matter intake and methane cannot be measured. So it's a good trait to consider in the breeding programs. And apparently, we have seen in this and other studies that theoretically we can achieve better genetic response for production, feed efficiency, methane traits, and overall profit, which at the end of the day is what farmers are interested in. And with that, I would like to thank you again for inviting me to present this seminar, and I can take any questions if you have some.
Thank you very much, Dr. Gonzalez-Recio, for your presentation. I would now like to open the floor to any questions, either from people here in the room or from those joining us online. We do have a question in the room. Okay.
Hello, Dr. Oscar. I am Emanuele. I'm a PhD student under supervision of Dr. Pavchenko, and I'm also work with the sniffer right now. We started getting data, so I'm really interested in this part. And I would like to ask if did you calculate the baseline, or how did you split which was the animal really phenotype and what was the environmental background air?
Yep. Thank you for the question. Yeah, that's an important part of calculating the methane phenotypes. I saw in the chat that Lucas put the date. It's April 29th. And we're going to explain how we calculate this in the different countries and breeding programs and scenarios. In our case, what we did is for each visit, we take the whole vector of measurements during the period that the cow is in the automatic milking system. We take the 5 lowest measurements and we average them. That's considered our background methane. And then we subtract that background noise from all the measurements that we get during that milking system, during that milking from each cow. So what we do is to calculate a specific background per cow visit.
Okay, thank you so much. I will get in touch with you in the future with more questions while I'm working with it, but I will watch the seminar from the other project. Thank you so much, Dr. Oscar.
Yeah, perfect. Thank you. Don't hesitate to send me an email with questions.
Other questions in the room? Oh. We do have a question.
Hi, Dr. Oscar. I had a quick question about your microbiome-driven strategies. When thinking about it from that perspective of selection, were you thinking about it in terms of abundance or diversity within the microbiome? What are you more inclined towards?
Yeah, thank you. Good question. Yeah, we're using relative abundances, actually. I know that some studies have used diversity, but at the end of the day, diversity is only one measure. We think that relative abundance are much more informative. And well, yeah, we have a lot of taxonomic groups or functional groups in the microbiome composition or in the metagenomics data. So we need to work with a system that reduces the dimensionality of the problem. There are several, or I would say like many approaches to do that. The one that we chose is to use these aggregated variables based on principal components. You can also use partial least square regression to calculate principal components, but then you're driving it from methane emissions. That's also an option. I think those are the ones that work better.
And that's interesting. Thank you. So you would say that even with archaea, considering we know that certain taxa, like Methanobrevibacter, are, I believe, if I'm not mistaken, really responsible for the majority of the methane production. Would you say in that respect as well, to focus on abundance instead of diversity?
Yeah, that's very interesting. If we will know the relative abundance of archaea, which are the only methanogenic microorganisms in the rumen, we should know, or they should be very correlated to methane emission, right?
Yeah.
So your question
Absolutely.
is very pertinent. And the problem that we see is that extracting DNA, well, it's several folds problem. Extracting DNA from archaea is not easy. So we cannot get the relative abundance of archaea like in a very representative manner with the kits that we use nowadays. And also there are different hydrogen sinks in the rumen, and not all of them are related to the relative abundance of archaea. So taking into account like the global picture, the whole picture of the rumen composition, we get a much comprehensive information of actual methane emissions than only looking at the archaea population.
Absolutely. All right. Thank you so much.
Hi, Dr. Oscar. My name is Samula, and I have a quick question for you. So, during when you're dealing with the data, did you have to work with different brands of sniffer, or even sniffer on the same brand but different generations? And if yes, there was any difference on the measurements or how you have to process that data?
Yeah, thank you for the question. So far we have been using only one brand of sniffers, but different settings, like different versions of the sniffers. We didn't see a lot of difference in terms of the sniffer, the different settings. Probably we used different brands. That will be a different situation. But with one sniffer, we didn't see many difference between the two versions. There is much more variability between farms than between sniffers or between series or version of sniffers. This is mainly because of the shape of the feed bin and the position, how we put the tube that collects the gas and the air in the automatic milking system. That generates a lot of more noise or variability than our sniffer versions in our case.
I see. Perfect. Thank you.
Thank you.
Any other questions? We do have a question online. Please, Dr. Lohais. We cannot hear
Think you're
you.
muted.
Sorry about that. Hey, Oscar, thanks for the nice presentation, and you're making some nice developments. When I saw your pasture index, grazing index, it made me think about some of that controversy around it was seen in sheep that in selecting for reduced methane, you're also selecting for smaller rumen sizes and higher passage rate. Does that cause a problem for the grazers? Trying to reduce methane, also, selecting for smaller rumens, higher passage rate, lower digestibility. How do you disentangle that? And can microbiome-driven breeding pose a solution to that?
Excellent question, Michael. I don't know, but that's a discussion that we have already. And I totally agree with you. And probably there is an optimum intermediate there that we don't want like too slow passage rate, but we also don't want quick passage rate because then the feed is not digested properly. There must be an optimum there, and I think that we need to work a lot with a nutritionist for this and study this more in depth.
And
Yep.
is it possible that for grazers, you might have a different microbiome that you select for?
Probably, yeah, I don't know. Probably yes, because they eating like a more fiber diet,
Right.
and they will probably need like a longer passage rate in the rumen. Yeah.
Right.
And where is the optimum? That's something that I don't know, but I think that's a good question. That's a good research question that we've been talking about that for a long time, and I think that at some point we need to put hands on.
Yeah, thank you.
Thank you.
We have another question in the room. Please, Ella.
Hi, Oscar. Thanks for a great presentation. I have two questions. One is about the collection of the microbiome information. Do you have any timing with collection of that data when you want it to be collected? I guess maybe relative to feeding or other information that might be relevant?
Yeah, that's another good point. When to do the sampling for microbiome, and it depends a lot if we are using rumen microbiome or if we are using buccal microbiome. For buccal microbiome, we want to obtain some bolus. So we want to wait until the cow is ruminating, so we can also get some bolus from the cow. For rumen sampling, ideally, we try to sample 2 hours after feeding, but that's practically that's impossible because we cannot sample all the animals at the same time. So there is always some differences in time to last feeding. We try to do this as short as possible, ideally 2 hours after feeding.
Nice, thank you. My second question is about the application of the microbiome data when you don't have methane or dry matter intake information. Do you see that more being implemented on farms where the management system isn't conducive to collecting those data points, or is it cheaper to collect the microbial information compared to methane or dry matter intake information?
Yeah, that's a good point. The cost depends a lot. I cannot say what is cheaper or more expensive because it also depends on the how many sniffers you have for the population. If you place the sniffer in a single farm, if you move them around and so on. Sampling microbiome is actually sampling is probably the most expensive part because you need someone that is well trained for that, and this is labor intensive, and you need to have a dedicated person that needs to go farm to farms. After that, sequencing is now very cheap, so our costs are under $100 per cow, DNA extraction and sequencing, and it's only we can do that once in a lifetime. So that's not very expensive, but comparing with methane emissions, it depends a lot. Definitely compared to dry marine intake is much cheaper because with dry marine intake you need to install weights or scales in the farm, and that's a huge investment in the farms.
Thank you very much.
Thank you, Ella. But I see you had a question in the chat, but I think Fatima ended up asking the same question, so I'm sorry for that. Is there any other question? Yes, Fatima would like to ask another question.
Sorry, I just had a follow-up question to Ella's question on when to sample. So have you noticed any, I would say because I believe there's some research that shows that certain animals that have higher or low methane production has a specific core subset of microbes that are present that is attributable to the phenotype of whether they have high or low methane. Would you say, have you noticed any interesting notes on whether there's a specific taxa or whatever that is related to a specific phenotype of whether the animal is high or low methane producing, would you say? Or not? I'm not sure that was clear.
Associated to time after feeding? Is that what you mean?
So because we know that the population that is in the microbiome is based off of what type of nutrition the animal is being fed. So I was just wondering if there's a core subset of microbes that are associated with high or low methane producing animals that you've noticed in your research.
Yeah, but this is not related to time to feeding. We have seen that there are a lot of eukaryotes, mainly protozoas and fungi that are associated to larger methane emissions. And there are also some bacteria, some prokaryotes like Prevotella, Butyrivibrio, and some others microbes that I cannot remember the names because they usually have strange names. I can share you next time, or I can send. Actually, we're going to publish this soon because what we can do is to calculate the loadings of each of the microbe or each of the microbe in the PCAs, in the principal components. So we identify what are the microbes that are more associated to larger methane emissions and lower methane emissions.
Okay, so you would say there's a subset in what you found.
Yep.
Okay, brilliant. Thank you.
Great.
Thanks.
This has been a great discussion. Thank you again for presenting for us today, Dr. Gonzalez-Recio, and thank you everyone for coming in person and online. The recording will be posted on YouTube, and I will share more details about that with you later. And that's all for today. See you all next week and have a nice weekend.
Thank you. Have a nice weekend. Bye-bye.
Bye. Recording? Hover over it. Here?